Actually changed. So it's pretty colorful for a colorblind organism, a novel mechanism for color vision. So I think we're all familiar with cephalopods. They're really cool. They change colors. Surprisingly, they're supposedly colorblind. So I'm going to spend the first little bit trying to convince you that this is a cool problem to try and solve. So here we are. These are 4 pictures of Sepia latimanus. Here you see it camouflaging in a red-rich environment. It is turning red. Here you see excellent camouflage in this sort of coral rubble with some orange algae. And I'll now show you a sweet video. So here, that was mostly backup in case the video didn't work. So here you don't really see anything. But there's the octopus. Okay. Can we do that again? Yeah. So you see it's perfectly the color of this green clump of algae here. You can't really see it. It's also mimicking the texture quite well. And then it blanches white because it's been discovered and it wants to starve. Then here, this is the same species, Octopus vulgaris, going over to a red clump of algae. It's not red yet. And then it turns red. So the question is, how do you do that if you're a colorblind organism? And I think we're presenting something that I believe is the answer to that. So they also break camouflage and they use chromatic signaling to conspecifics. It's difficult for me to envision a selective scenario where you would have highly conspicuous chromatic signaling if you were unable to see your own signal. Here's another cool video of cuttlefish. Or, does this work? Never mind. So there would be a really cool video of these cuttlefish signaling to each other. This is Sepia apama on the bottom. Basically, they have incredible displays that they use when mating, and here you can see it in big fin reef squid. Point is, they're very chromatically complex organisms. They signal to each other in color. It's difficult to imagine that a soft-bodied mollusk that's delicious to everything would signal to its conspecifics in color if it could just synthesize a similar luminance function in brown and white, for instance. So, yet the current scientific consensus is that the cephalopod visual system is colorblind. So why is that? It's because all previously known mechanisms for color vision require multiple photoreceptor types, and cephalopods have only one. The assumption is that without multiple photoreceptors, organisms can't determine spectral content of their image. And despite extensive research, all except potentially one organism, the Japanese firefly squid, have only one photoreceptor type, and they fail certain tests for color blindness in the laboratory setting that I'm going to argue are tests designed primarily for the human or standard opponent model of color vision. So here's a New York Times article to just show you that it's an interesting problem. Dr. Roger Hanlon is one of the world's experts in this, points out that they need to be able to figure out how to measure the color blocks in order to accurately camouflage. And the current hypothesis is that they're doing it in some magical way that we don't yet understand. I think the issue is, this is also brought up by intelligent design proponents as such a design speaks of a designer who gave animals what they need to survive and did so with some techniques that we are still struggling to understand. Everywhere we look, God's wonder-working hand has gone before. So this is the other alternative hypothesis. Yeah, untestable. So I'm going to say that I think that you can solve this with physical optics. And most refractive indices induce chromatic aberration. Different lights, wavelengths, bend differently. So you see this in a prism where you have short wavelength bent more. And you also see this in the radial gradient index of refraction in cephalopod lenses. Here, for both, dispersion causes blue light to come to be bent more and thus comes to a focus at a different point than red light. Interestingly, the image quality budget in these organisms is also dominated by chromatically induced blur. We went through and calculated the different sources of error in cephalopod visual systems. Fortunately, we know very well what size their photoreceptors are, estimate the retinal surface errors, and residual spherical aberration. Turns out chromatic blurring is, for almost all pupil shapes, the dominant image term. But they actually have these angular pupils in cuttlefish and reef squid in particular, which maximizes the chromatic blurring. So chromatic blurring is minimized if you have a small aperture, which allows the light rays to come to a cone, and any displacement of the focal plane doesn't induce much chromatically induced blurring. But if you're imaging off-axis, in effect, in high light, they're maximally maintaining the #1 source of image blur. So I think that's pretty cool. So here you see the off-axis imaging system that's in cuttlefish and reef squid eyes. All right, so they predominantly focus through colors and not actual object distance due to chromatic aberration. So here you see through a simulation, if the distance to object in meters is beyond 1 meter for a sort of 1 meter sized cuttlefish eye, they're actually focusing through colors. So the accommodation point of best focus at 550 nanometer monochromatic light is quite different than the best focus for blue light. So you have this sort of color range ambiguity, where if you know something is in best focus at this accommodation setting of 12.2 millimeters, the object is either 1/3 of a meter away or 2 meters away if it's red. So you just have to be able to break that ambiguity to be able to determine the spectral content. And we know that image defocus is used in distance judging by chameleons and jumping spiders. So I think this is computationally a very possible thing for these guys to solve. So we did a computer simulation of chromatic aberration in MATLAB, where you create a test pattern bar chart. We used real fish reflectance for tropical reef fish, and then each assign the spectral content to each pixel, weighted by opsin response and filtered sunlight attenuation through seawater. Select a pupil mask, and then you create a hyperspectral image cube, 1 plane per wavelength, and then you pick focus settings, and we can go through and see it cool. So here you're going through best focus, and this is a yellow pattern, so here this MTF is maximized, and you see it in good focus when the accommodation setting is best focus for yellow light, and that is out of focus here. So here you see, all right, so here we'll see it then, goes through, gets to slightly better focus, then at about 600 nanometers or so, it'll start falling off again. So here, the point is, they don't see all colors in focus very well at once. They pretty much only are able to image a particular wavelength as being in good focus at a particular accommodation setting. So by doing an accommodation scan, that effectively is a spectral scan of the scene. If you do those spectra from blue and yellow reef fish, you see at blue best focus, a blue and black test pattern is very well resolved. At red best focus, this is not particularly well resolved. These line cuts is a contrast transfer function to be able to measure image acuity in these organisms. And you'll see that with the annular pupil, this blue is the contrast transfer function, and the red line is the photons that would be detected over the integration of the opsin response and attenuation due to seawater. So you'll see that the CTF, so best quality image, basically tracks the detected photon intensity in these test patterns. And interestingly, on blue and yellow colors directly adjacent to each other, you can't get the sign of defocus, because as one wavelength of light goes into focus, the other defocused image from the adjacent pattern means that you never actually resolve that edge. So I think that explains why these standard optomotor blue and yellow patterns rotating around the animal to measure nystagmus response fail to detect color vision in these organisms. All right, so then the off-axis pupil shade greatly degrades visual acuity. If they just had a dot pupil like we do, this CTF is pretty uniform, it's very good. But they don't. So they're really taking a huge hit on visual acuity in order to maintain this ability to make spectral inferences from the off-axis imaging system that they have. So here's another cool image. This is a ray tracing result through the annular pupil, where you can see that it's a deterministic output, whereby wavelengths greater than 400 nanometers fall out on one side and less fall out on the other side. What happens is as it moves through an annular pupil, there's a parity flip, and that allows you to be able to see the sign of defocus. So if you're imaging a monochromatic point source and it's really blue and you're at too red of a focus, it'll appear in one conformation, and then when you go out to the other side of defocus, you have a parity flip there. So the implications of these modeling results are that organisms with only one photoreceptor type can extract spectral information through the exploitation of chromatically induced defocus. And extracting color information from the CTF versus accommodation requires crisp changes in intensity. You can't do this sort of imaging system if you have no network of sort of fine black lines. And color to black transitions work best since this minimizes the crossover out of focus light from the other side. And we also note that cephalopod displays almost always have sort of color washing over a fine network of black lines, which is sort of the perfect scenario for measuring crispness of an image. So much as you could think of this as analogous to the autofocus mechanism in a digital camera, which works on the same principle. It seeks the focus setting that maximizes the high frequency Fourier component in the scene and would have difficulty focusing on a featureless flat monochromatic scene. You also see this in cephalopod behavioral experiments. They fail to color match if you put them on a red piece of construction paper. Then if you give them a naturally textured background with focusing aids, they succeed at color matching. So I think this is also applicable to a number of other organisms that maintain these low F-number visual systems with off-axis pupil shapes. Dolphins have only one cone type. They have a similar U-shaped pupil. And there's behavioral evidence for spectral discrimination when you have focused point sources in spectral regimes where their visual system would have difficulty encoding color through opponency. Many spiders also have only a UV-sensitive and green-sensitive opsin, but they image across 3 to 4 focal planes simultaneously. Chromatic aberration, we also know, is the leading source of image blur in these organisms. There was a really elegant paper, Nagata et al., that showed that the jumping distance for jumping spiders varied depending on the illumination and light, so we know they use image defocus for calculations of distance. This tiered retina is probably the optimal morphology for measuring this chromatically induced defocus, because rather than having to go through accommodation settings, they're in effect measuring it across 4 accommodation settings at any one time. And it's also interesting that these crab spiders have an ability to background match. Jumping spiders' preference for colored mate disappears under fluorescent lighting, which is a series of line emissions that would break this model, and we have simulations to show that as well. So this organism is red-green colorblind. It has only a UV and green opsin. Looks like it's got a lot of red and green to us. I would argue that it's difficult to imagine that this sort of a scenario could evolve under a selective pressure where all of their predators can see red and green, but they can't. Why wouldn't you just have a less conspicuous signaling mechanism? So thanks to Jim McGuire, my advisor, for advising in my career in biology. Sonka Johnson provided really great comments on this manuscript. Marty Banks at Berkeley and Vision Science also provided excellent comments, and the whole McGuire lab. The first time I gave this talk, I had no funding, but now the Museum of Vertebrate Zoology ironically gave me funding to study cephalopods.